Good morning, everyone. It is Tuesday and January the 12th. It's 9 a.m. here in Cleveland, Eastern Standard Time. My name is Ashok Agarwal. I'm your host for the next 2-3 hours. And I want to, first of all, before we begin the program, want to wish you a very happy New Year 2021. I wish you good health and a lot of happiness, and I hope that we will be able to see many of you during this year after the pandemic is under control. So thank you again. And today is a very exciting program. We are here to discuss and present virtually our online ART training. Today is the third program in the series. We began in November. We presented first program in November, then second one in December, on December 8th. And today is our third program on January the 12th. Today's program is on ancillary semen test. So let me begin without taking any further time and begin my introduction. Okay, very briefly, this is my biography, a very brief biography, just to provide few highlights. I am in charge of the Andrology Center at Cleveland Clinic for the past 28 years. I'm the director of the American Center for Reproductive Medicine, which is the host of this online virtual training session. I'm a professor of surgery and urology at the Lerner College of Medicine at Case Western. I'm a staff in urology and in some other department. Our group is very well known in the area of male infertility, andrology, and human-assisted reproduction, and we are ranked as #1 in terms of our publications based on the Scopus, which is one of the premier service in terms of the literature in the medical field. Our group has published a very large number of books in the field of male infertility, ART, fertility preservation, DNA damage, and antioxidant. Our laboratory has trained over 3,000 scientists, clinicians, graduates from more than 100 countries. The organizers and speakers of this program have no conflict of interest. We have taken no sponsorship or asked any company for that. This program is made entirely possible by the support from our foundation, from our own center, and the webcasting support has been provided by our colleagues, David Reichling and David Wolfley. Course faculty have generously donated their time for this online training, and our program coordinators, who you will see in a minute, have graciously volunteered their valuable time to manage this program. Lastly, the names, images, and description of products or instruments that you may see during the lectures or which will be discussed during the presentations are not being endorsed. They are being used routinely in our program. So let's talk about the genesis of online ART training. Some of you have been listening to this. We got this picture actually drawn by our medical artist for a publication that we are planning to do. So essentially, the program was created because of the pandemic to offer the highest quality educational experience in human-assisted reproduction on a global level using online content delivery model. And we wanted to make sure that it is free, so anybody who's interested in this field is able to join. There is no fee. Our goal is, our aspiration is to raise the bar on quality of lab skills and theoretical knowledge in both the field of andrology, embryology, and overall in assisted reproduction. We feel that the people who work, the reproductive professionals who work in these laboratories are the foundation of what the patient receive in terms of the care later on. So we have to make sure that we provide the best training to our colleagues all over the world who are working day and night in taking care of multiple work related to the diagnostic and services in human-assisted reproduction laboratory. Without them, the entire field of assisted reproduction will not rise to the level that we would like to see and provide the best care to our patients. We have, for today's program, participants who are joining us from 64 countries. We a little bit of details about our programs. We have 8 modules which are going to be offered in 14 virtual sessions. No registration fee, no any fee. Certificate of participation are issued by our program and endorsed by the College of Embryology here in the United States. We have experts in andrology and human-assisted reproduction who serve as faculty for the program that we are providing. We have lectures, multiple choice questions, and sharing of important reading material and technical protocols provided to every single person who has registered. We started with routine semen analysis. We are still part of routine semen analysis, which are the ancillary tests. We are going to move next after this program into advanced sperm function test. Then we will move into ART, embryology related lab procedures, and essentially complete the circle in the next several months. The criteria for certificate of participation is through multiple choice question. We have anywhere between 50 to 65 questions from the content covered in each online module. And the candidates, applicants have 20 to 30 seconds to answer each question. Each person must pass the test if they score more than 65%, 65 or greater than 65% correct answers. We are still debating raising this score, passing a score to 70% or 75, and this is still under consideration. The online survey, attendees will be asked to fill out an online survey. This will be at the end of each module and after the MCQ. Survey results will be used to improve the organization of the next module. We take it very seriously. Look at each and every response that we receive from all of you after the course is finished, and fine-tune our program wherever we feel that we have not provided the best service. This is the highlight of the last module, Module 1B, which was devoted to morphology. We had more than 900 delegates from 58 countries who finally attended the program. Of course, there were a lot more who registered, as you can see, almost 1,200. So the program was very successful. About 826 or 92% of people took the online test, and 69% or 570 candidate pass the online test and they qualify to receive the special certificate, which we were showing in the PowerPoint display just before we began. These are our program coordinators, as you can see here. Dr. Manish Kumar, Dr. Renata Finelli, who are working at Cleveland Clinic. They are essentially doing the things non-stop every single day. Camila Pompeu from Brazil, Neha Singh from India, Masha Darbandi from Iran. Further, we have Andrea Bello from Brazil, Sara Darbandi, Iran, Raha Sadeghi from United States, Sarah Madani from Algeria, Simran Kovaribus, sorry, my last name may not be correct, from the United States. These are the highlights of our online ART training journey. If you look at module 1A, we have about 1,337 candidates. For 1B, more than 1,451. And for module 1, which is today's program, we have about 1,150 candidates as of today. So some summary statistics. The very first program, which is module 1A, we had a yellow bar shows the number of total candidates. The orange bar shows the number of those who took the test, and you can see the green bar is very close. We had a very high rate of passing. That means people are really enjoying. They are doing very well. They are listening to the speeches and talks and presentations. And this reflects in the very high rate of passing the multiple choice test. So we congratulate each one of them, as well as we encourage today's participant to do very well, and good luck to them. And this is the data from the survey, and the registration. The top 10 countries who have registered, you can see in each of the modules. So the top country remains from India, then Brazil, then Egypt, Nigeria in the latest version, which is module 1, which is on the very right. So this is, and then we have a host of other countries such as Iran, Indonesia, Pakistan, Mexico, and so forth. For a complete list of participants in today's program, you can see these are the candidates who have registered from 64 countries. And these are some of the certificates that, this is just some examples of certificates, special certificates that we have been issuing. You can see each certificate is customized, personalized with the name and pictures, professional pictures of each of the candidate, printed with their degrees, the city, as well as the marks that they have scored in the multiple choice test. And we certify what course they have taken, and the training is endorsed by the American College of Embryology with the signature of the president of the American College of Embryology, my good friend, Dr. Dimitri, and also certified and endorsed by our program in Cleveland Clinic. These are some of the examples of the individuals that we just put here to encourage you to take the multiple choice. So this is today's program. The learning objectives today is we have a very, very good program today, and you will see this is part of the semen analysis, and it is like a hidden gem. Most people will not know many of these tests that are basically done as part of semen analysis as indicated. So we are going to discuss about the Leukocytospermia, or detection of white blood cell or infection, sperm vitality testing, anti-sperm antibody screening, special test for Isospermia, post-vasectomy screen, and screening for retrograde ejaculate. So I can tell you that today's program is by far going to be the very best. We have invested enormous amount of time in preparing for these lectures. They have been well researched, well prepared, and practiced dozens and dozens of times what you are going to see today. So for each lecture, we have spent almost 30, 40 hours minimum in practice sessions. And each lecture has gone through at least 10 or 12 practice sessions with multiple people. And each content has been well researched through the literature with proper references, and to make sure everything is extremely accurate and simple for you to understand. So this is our program today in a diagram. As you can see, the last lecture is going to be by our clinician, who's going to really put it all together to explain what these tests really mean to you. What shall we do with them? And our line of speakers today are my colleagues here and my good friend, Professor Ralph Henkel. You can connect with our program by connecting on our LinkedIn, and these are the benefits. Almost every single day, we provide update on our research activities by posting hundreds of research articles freely, completely downloadable, that anybody can read. We also post the latest research which are presented at prestigious meetings such as ASRM, ASHRI, and AUA. You can learn about our virtual research opportunities, which we have just started. You can learn about our online training and opportunities to collaborate with our team. Just go to the LinkedIn and send a request to Ashok Agrawal, and it will, from Cleveland, and it will come to us, and we will allow you to connect to our program. There is, okay. Our first speaker is Professor Ralph Henkel. He is a highly decorated researcher, academician, and a teacher, all rolled into one. And we have been very, very good friends, but he is listening to us, and he has recorded his presentation just to avoid any kind of technical problem. But let me come back to his biography, which is very, very impressive. As you can see, currently he is a visiting reader in the Department of Metabolism, Digestion and Reproduction, a mouthful, in the Imperial College of London in the United Kingdom. He is also the chief scientific advisor at a very important company called Logix Pharma in Reading, United Kingdom. He essentially moved from South Africa, where he is currently an extraordinary professor of reproductive biology at the Department of Medical Biosciences, and in a host of other places. He is also affiliated with our program for many, many years. We are working together. Most importantly, Ralph is the editor-in-chief of Andrology, one of the original and most prestigious journal in the field of andrology in the world. He is very well recognized in the world, and you can see all his information in the slide that we will later on share with you.
Good morning, good afternoon, good evening, everybody, and welcome to my presentation on Leukocytospermia today. The learning objectives today are to understand the need to examine the presence of round cells in semen, testing the presence of white blood cells in an ejaculate, defining a leukocytospermia condition, and how to test for leukocytospermia. And of course, when we test clinically, when we need quality control, and we also need to understand the relationship between leukocytes and an infection. What is a leukocytospermia? Leukocytospermia is defined according to WHO in the latest manual of 2010 as a white blood cell number of more or equal 1 million white blood cells per milliliter of semen. This is a condition that is present in 10 to 20% of infertile men, and it is an indication of male accessory gland infections, or abbreviated MAGI. And this is or can be seen in up to 15% of male infertility cases. Where does these infections take place? That is mainly in the testis as an orchitis, in the epididymis as an epididymitis, prostate as a prostatitis, or in the seminal vesicles as vesiculitis. And that goes along with swelling of the relevant organs here, and real infection of these organs. It's getting along with fever, temperature, heat of the relevant organs. It's painful, and it's due to a number of pathogens like chlamydia, ureaplasma, mycoplasms, enterococcus, staphylococcus, and pseudomonas. The detection of these conditions is difficult because in many cases, these patients are asymptomatic. We also call that as a silent genital tract infection. And we need to understand that these MAGI, or these male accessory gland infections, they contribute to increased seminal leukocytes. And that is in the end leading to increased production of reactive oxygen species, and as a result of that, to failed fertilization and pregnancy. And then we have problems with treatment of the antibiotics, because many of these pathogens in recent years became resistant to antibiotics. And that must then be tested in a semen culture where the different possibilities can be checked for the sensitivity to different antibiotics. Also, there are different types of pathogens available, aerobic and anaerobic, and both aerobic and anaerobic pathogens are present in semen, with anaerobic pathogens even in almost every ejaculate available. In addition, there are different other challenges that need to be observed. There is no agreement and no guidelines from the WHO, from the AUA, ASRM, or from the European Association of Urology to give guidance when a semen culture should be done. In addition, there is the relevance and significance of the positive semen culture is debated, but on the other hand, it's critical to distinguish between semen infections and to know what is going on there for an effective management and the way to treat these patients, so that we know what can be done. In this recent study of last year by Burry, these authors investigated more than 1,600 patients. And among these patients, they found asymptomatic, so with patients with a silent infection, and a positive semen culture that was 21%. It's a substantial number. And most of these pathogens that were found was ureaplasma, urolyticum, and there was enterobacteria, chlamydia, mycoplasms, and others, even including a virus. And in more than 10%, there was even more than one agent, one pathogen detected in the semen. As a result of all this study, these authors could clearly see significant differences for sperm count, progressive motility, and not between positive ejaculates with the infection and those that are not infected. In another study, Ventimiglia, also from last year, these authors investigated more than 500 patients. In 90% of these patients, the semen culture was negative, However, in 10%, the semen culture was positive, and if they analyzed these 10% further, only 20% of these patients were positive for Leukocytospermia. That means that 80% of these patients would not have been detected if they would just have relied on a Leukocytospermia test and would not have done a semen culture. So that means that Leukocytospermia is not necessarily a good predictor of positive semen culture, and it's no single parameter is sufficient in medical decision making, because we need to treat the patients properly and effectively in the end. What kind of leukocytes will we find in semen? It's the majority of the leukocytes in semen are granulocytes. That makes between 15 and 60%, and that is neutrophiles or polymorphonuclear granulocytes. Then there's about 25 or 20 to 30% of macrophages, and the rest is then T and B lymphocytes. And the detection of these leukocytes in semen, there's different methods available. We can do it with immunocytostaining chemistry, that is regarded as a gold standard, with monoclonal antibodies against specific white blood cell subpopulations. And this can also be done in flow cytometry, as shown here on the right-hand side. You can clearly distinguish here this group of leukocytes, and here at the bottom there are the spermatozoa. On the other hand, there's specific tests available, like seminal granulocyte elastase test, and this is done in an immunoassay, ELISA. But all these tests have limitations. They are, number one, expensive, they are time-consuming, and they are not standardized. So what can be done? There is routine tests that give an indication what is going on in the ejaculate. In a wet prep, one can see round cells, but these cells cannot be really distinguished from immature germ cells, and it's highly inaccurate. So we need to do other techniques, including staining techniques with Papa-Nicolau or modified Bryan-Leishman staining. But again, these techniques are time-consuming, require really very specifically trained staff in the laboratory, and when they are not precise. And it's a problem to distinguish between different germ cells and different round cells and leukocytes. So what is left, and also been recommended by the WHO, that is the orthotoluidine blue test and the ENS test. What we see if we look in the ejaculate in a normal light microscope with our round cells, that is for the initial semen analysis in the wet prep. And what we then also see that we cannot distinguish between leukocytes and immature germ cells. There is even other cells available, epithelial cells or trichomonas, and these cells here, primary spermatocytes, here is spermatid V, here even the trichomonas. You cannot really distinguish between these specific cells and is it active leukocytes. So we need to get down to the bottom and clearly identify leukocytes. And that, but this examination for round cells, that is a first indication. And if there are more than 5 round cells per high power field, then we need to check for white blood cells or leukocytes. If we see more than this white brown blood cells, and that should be done according to this formula, we count in a light microscope all these 10 by 10 grids, and it's more or equal than 1.0 million leukocytes per milliliter, then we have to do the leukocyte test. And there are two different leukocyte tests available. That's an ENS test or the peroxidase positive cells test with the orthotolidine. Like I said, that test should be done if there is more or equal than 1 million round cells per milliliter ejaculate, when either ENS test or peroxidase test. The orthotolidine peroxidase test is recommended by the WHO. We need to prepare all these chemicals, reagents, here working solutions. That is when must be done on a daily basis. After staining procedure, what is shown here, when we see in the microscope peroxidase positive cells, that is those leukocytes that still have the peroxisomes in the cell. They are staining brown like these cells here, and those that are negative, not leukocytes, that is these cells here, they are markedly lighter and more whitish. On the other hand, there is the ENS test available, which is named according to the person who first described this test in 1974. We also need to prepare all these stock solutions, which are stable for about 6 months. And then the working solution should be prepared on a weekly basis. The test is then performed with 20 microliters of the ENS reagent, with 10 microliters of thyroid buffer or PBS buffer, and 10 microliters of liquefied semen, which is mixed in an Eppendorf cup, and that must then be a dark brownish one to prevent it from exposure to light. After the incubation, we can analyze it in a macro chamber under a bright field microscope, count the peroxidase positive cells, and do the calculation. And this slide shows you the performance of the ENS test and all the equipment that you will need. And now I'm going to show you a small little video about the performance of the test. Here you take the 20 microliters of ENS solution in a dark Eppendorf cup. You add the PBS of the solution or thyroids, mix carefully, and then add the working solution. Mix again, and then incubate for 5 minutes. Afterwards, you prepare your microscope with a bright field. Maybe take away the filter. It is used with a 10 times objective lens. The macro chamber will be filled with the exposed and incubated solution with the sperm. Now the cover slip comes on top, and we can then analyze this in the microscope. Of course, for any normal diagnostic test, we need to do the quality control, and that should be done on a weekly basis for the ENS test. The ENS positive control should be run, and the result should be more than or equal 1 million white blood cells per milliliter. If the results are less, then prepare new reagents and retest the control. If there's no leukocytospermic sample available, one can also use an EDTA anticoagulated blood specimen. And here on the right-hand side, I'm just showing you some examples of a quality control sheet. Here you can see that the sample that was done last year in September, 7.6 million leukocytes per mil in the old solution, and the same here for the new solution. So that is perfectly correct, and there is no difference between these. So that quality control test is positive, and that's in order. What kind of leukocytes can we detect with the ENS test? We detect neutrophils, eosinophils, and basophils, but we cannot distinguish between these three different types of granulocytes. Again, if the result shows less than 1 million leukocytes per milliliter, that is then not leukocytospermic. If it's positive, it's more or equal 1 million leukocytes per milliliter of semen. Where are these reactive oxygen species coming from in semen? That is, of course, here from white blood cells. That is their normal function, what they are doing to actually attack pathogens and destroy pathogens. On the other hand, they are also coming from abnormal spermatozoa, and we must also be aware of the fact that the activated leukocytes, like here, they are producing roughly 1,000 times more reactive oxygen species than normal spermatozoa. And how is that done? In abnormal sperm and in leukocytes, they follow basically the same biochemical pathways. When apoptosis is activated and oxidative stress via reactive oxygen species and reactive nitrogen species are released, which in the end damage the DNA, proteins, and the lipid membranes of good quality spermatozoa, leading to a reduced fertility of that specific patient. If we have now leukocytes in a semen, that is an indication there is also more reactive oxygen species produced and released, and this is we can clearly see here in this study, Agarwal on 2014, in leukocytospermic samples, the production of reactive oxygen species is significantly higher than in semen samples with no leukocytes or even in non-leukocytospermic patients. This elevated level of leukocytes has also a significant effect on sperm motility. So motility goes down in semen samples that contain higher numbers of leukocytes. As well, here in this study, the amount of DNA damage is increasing significantly with elevated numbers of leukocytes. And here in leukocytospermic patients, it's also significantly higher in this other study by Saleh et al. And with this, I want to provide the take-home message. The peroxidase staining by ENS test or the toluidine blue stain, while it's not a gold standard, it's simple, it's practical, and it provides a reliable result. Leukocytospermia is called when there's more or equal 1 million of white blood cells per milliliter of semen. It may indicate an underlying genital tract infection in terms of MAGI, and leukocytospermia is not a good predictor of a positive semen culture. That's why both tests, the leukocytospermia, the detection of leukocytes with the ENS test also, and the semen culture should be done in parallel. and Leukocytospermia is a significant factor associated with high reactive oxygen species, resulting in poor sperm quality and DNA damage. And with this, I thank you very much for your attention. Here are some pictures from our group. And this is where I'm staying at the moment in London.
Okay. Thank you very much, Ralph, Professor Henkel, for excellent presentation on a topic of great interest on leukocytospermia, or presence of white blood cells in semen. There is a lot of a story behind this infection, and it's quite complicated. So he tried to explain it that the test for leukocytospermia is really not a great test to tell whether the culture is positive or not. So now we dive into the next presentation, which will be given by my associate, Dr. Rakesh Sharma. And Dr. Sharma is an associate professor at the Lerner College of Medicine at Case Western. He is the coordinator and assistant technical supervisor within our Andrology Center. He has been with us for over 26, 27 years, and he has done a lot of research. As you can see, he has received several awards, and he is an excellent andrologist in his own right. So today, I have asked Rakesh to present this presentation on sperm vitality. So let me begin, and I'll ask Rakesh to begin his presentation at this time. So Rakesh, you can begin at if you are able to.
Thank you, Dr. Agarwal. Hello, everyone. The topic of my talk this morning is sperm vitality. Next slide, please. The learning objectives will be to understand the differences between sperm vitality and sperm viability, learn step-by-step protocol to conduct this test, understand the quality control associated with this test, define the normal and abnormal results, and look into the clinical correlation and the therapeutic application of the sperm vitality test. And at the end of my talk, I will provide you with some important takeaways. Next slide, please. It is important to differentiate between live, non-motile sperm and dead sperm. And also, it is important to understand the difference between sperm vitality, which essentially refers to the integrity of the sperm membrane, and sperm viability, which refers to the percentage of live sperm in the semen sample. Both terms are frequently used and interchanged, but I want to highlight that this is not correct. Both terms, they are different and distinct. The only common thing between the two terms is that they both indicate the function of the sperm membrane. On to your right is the protocol for conducting this test, and this protocol was shared with all of you. Next slide, please. The test for sperm vitality is the eosin-nigrosin staining, and this must be conducted on all specimens having a motility of equal to or less than 40%. And this test is done immediately following the initial examination of sperm motility. And this is important because any delay in conducting this test can result in deleterious effects because of the dehydration or changes in the temperature. Eosin-nigrosin is a common test, but there are other tests available such as the trypan blue, propidium iodide, and the Hoist stain. Again, on the right is the protocol or the brief protocol that was also shared with all of you. Next slide, please. The principle of the eosin-nigrosin staining for sperm vitality is based on the cell membrane exclusion, principle. And what this means is that if the sperm is intact, the stain cannot penetrate the cell and the sperm will appear white. However, when the sperm is damaged or the membrane is damaged, the stain starts to enter the sperm and the sperm head will stain and eosin will be shown in the sperm as a pink color stain. And on to the right are the stains showing the vital sperm, which is not stained, appears white, and the stained sperm, which appears pink. Next slide, please. It is important, as I mentioned, to know that the sperm which are immotile, are they alive or dead? And this is examined after the motility has been done, and the presence of large number of vital but immotile sperm, they may be indicative of structural defects in the sperm flagella, or this is also called the ciliary dyskinesia or Catagner's syndrome. And the image on the right top shows the 9+2 arrangement, which is seen for the cilia as well as for the flagellum. And the outer 9+2 arrangement shows the dynein arms, which are highlighted in green, and these are the outer and the inner dynein arm. In the image below is the image of an electron micrograph of the flagella showing a normal 9+2 arrangement, and the other three images show when the dynein arm is absent, either both are absent or one outer or the inner arm of the dynein is missing. And a high percentage of immotile and non-vital cells, they indicate necrospermia, which is also an indication of an underlying pathology responsible for the non-vital cells. Next slide, please. This slide shows the preparation of the stains, and essentially it contains two stains, the eosin, which is a 1% solution, and nigrosin, which is used to add contrast. It is a 10% solution. Next slide, please. This slide shows some of the staining supplies, and the image on to the right shows a special porcelain slide, which is a 3-well slide. It's known as the Werner slide. Next slide, please. And this is the protocol where we place 1 drop of a well-mixed semen sample on the Werner slide, and we add 2 drops of eosin Y. We mix the sample and add 2 drops of nigrosin, and immediately a smear is prepared, air-dried, and ready to be observed under bright field microscope. Next slide, please. Here I would like to show you a video clip as to how this test is conducted. So we prepare the slide by providing the patient information, and we mix the sample on which the motility has been examined. We take 1 drop of a well-mixed semen sample and place it in the Werner slide well, and we add 2 drops of eosin stain and we mix it well. This is followed by 2 drops of nigrosin, which provides the contrast. And nigrosin is a very dark stain, so we have to be extremely careful that the stain is placed right in the sample and not anywhere else. The sample is mixed thoroughly, and then we take 10 microliters of the sample and place it on a clean glass slide, and we prepare a thin, uniform, feather-like smear. The slides are allowed to air dry, and they're ready to be examined. Next slide, please. The microscope setup includes the bright field microscope. We also make use of a 100x objective and a 10x eyepiece. The immersion oil used should be of a high refractive index, which is 1.52, and we also use the differential counter, which is shown on the image in the right, to count the viable and to count the stained and the unstained spermatozoa. Next slide, please. This slide shows that the sperm which are live, they will appear white, whereas the sperm which are dead, they will appear pink. Now, on many occasions, we will see a small portion of the neck region which is stained. This is called as a leaky neck membrane. And it is important to know that this is not a sign of cell death, and these sperm are scored as live cells. The WHO 2010 reference range for normal vitality is equal to or greater than 58% of the sperm in a normal sample. And this is an important diagnostic test in patients who are asthenozoospermic, or those patients where the motility is less than 40%. In general, the vitality should be equal or greater than motility. On the right, you see two images of a viable cell and a non-viable or a dead cell. Next slide, please. Here I want to illustrate a scenario. And in this case, a patient provides a semen sample, and upon examination, it is noted that the volume of the sample is very low, 0.3 ml, and it also has a very low concentration or oligospermic sample. The motility observed is 15%. The sample is spun and stained for the eosin nigrosin. When we score the slide, we find that the average vitality is 9%. Now, the vitality results are lower than the motility results, and we need to know what is the reason for this, because we explained that the vitality results should be equal or greater than motility. So in this case is an example of a poor quality semen sample where, number one, the semen volume is very low. Number two, the concentration is also very low, and the sample is spun. And that is the reason why this result may be inaccurate. And this inaccuracy can be seen in sperm with poor quality. Next slide, please. The next test for viability is what we call the hypoosmotic swelling test, or the Haas test. And this test is done to check the viability of the sample without staining. And the results of this test show the swelling of the tail. The reference range, the reference value according to the WHO 2010 edition is equal to or more than 58% of the sperm which show tail swelling. Now, I want to illustrate to you two scenarios which describe when we should use a eosin nigrosin stain and when we should use the viability or the Haas test. Scenario number one is if we are conducting a diagnostic test and the semen sample has less than 40% motility, we make use of the vitality test or the eosin nigrosin staining. The second scenario is when we want to use the sample for therapeutic purposes such as in ICSI. In this situation, the motility is 0%, and we will use the Haas test, which is not staining the sample, so the same sperm can be injected, and the swelling of the tail will confirm that this non-motile sperm is viable. Next slide, please. As in any test, quality control is the hallmark of the efficiency of conducting the test and the validity of the results. So in this case, we have to also perform the quality control, and this involves the patient sample that is used to check the quality of the reagents. The motility is checked before checking the reagents, and this is conducted by using a new lot of the reagents and comparing with the existing lot to make sure that the new lot is functioning correctly. The vitality with the two lots should not show a difference of more than 10%. On to the right is the quality control sheet, which shows the lot number and the expiration date of the two dyes that we use, which is the eosin and the nigrosin stain. Next slide, please. The acceptability of the results is very important. And here I would like to give you another scenario where we count the sperm and we find that the average of the two readings is 17%. The two sets of readings are 22% and 12%, so the difference of the two readings is 10%. Are these results acceptable? On to the right is the table from the WHO 2010 edition, which shows what should be the average and what is the acceptable difference. So from this example, if we look, the average reading is 17%, and the difference that is acceptable is 8%. However, in our scenario, the difference in the two readings was 10%, and therefore, these results are not acceptable, and this slide should be rescored. Next slide, please. This is another scenario I want to illustrate where the two sets of readings were 30% and 34% with an average of 32%. And the difference in the two readings is 4%. Are these results acceptable? Again, if we look into the table on to the right, we find that for an average of 32%, the acceptable difference is 9%. And this is matching with the difference that we are getting, which is 4%. And as the difference is less than 4%, these results are acceptable. Next slide, please. Sperm vitality is an important measure of sperm fertility. Also, we find that the normal sperm vitality is highly correlated with the normal DNA integrity. And this was documented by a study done by Samplaski and investigators, where they were examining the results of sperm vitality and the sperm DNA integrity. And they reported that normal sperm vitality is highly correlated with DNA integrity. And abnormal sperm vitality is correlated with a high DNA fragmentation. And this is illustrated on the image to the right, which shows the inverse correlation of sperm vitality with DNA fragmentation. And this correlation is very strong, negative correlation, as you can see in the box on the right. The correlation of 83% indicates that the sperm vitality is a good marker and that can serve as a surrogate marker for sperm DNA integrity. And this is important for those labs which are not equipped with sophisticated equipment. And in those situations, the eosin nigrosin stain can be used as a marker of sperm vitality. Next slide, please. So I would like to leave you with the take-home message, and that is that the sperm vitality can be examined by the eosin nigrosin staining, especially in samples which are of poor quality. Eosin nigrosin staining is simple and accurate test, and the vitality results must meet the criteria for acceptability before these are reported. And the vitality results correlate highly with both motility as well as the sperm DNA fragmentation index. And viability can be examined by the Hoss test, and this test can be used for therapeutic purposes such as in ICSI. Next slide, please. So I want to leave you with a group picture of our ACRM team, and thank you very much for your attention.
And now we dive into the next topic, which is on anti-sperm antibodies. And our next speaker is Dr. Sejal Gupta. Dr. Gupta is a veteran of our andrology program. She is the technical supervisor of our center, which runs 3 laboratories in different parts of Cleveland Clinic, in different cities. And also we have a very large reproductive sperm and semen bank for cancer patients. Dr. Gupta has a distinguished career. She has done her fellowship in reproductive medicine with WHO, and then a research fellowship with our program. She has been highly decorated, as you can see. She has also done a master's in embryology from the Jones Institute. She is a very passionate andrologist who really takes care of our patients in the most compassionate manner. And today I invite her to speak about the issue of anti-sperm antibody test. So Sejal, you can present if you want.
Thank you very much, Dr. Agarwal, for the introduction, and welcome to all the participants. Good morning from Cleveland. And the presentation that I'm going to be making is on anti-sperm antibody test. Next slide. The learning objectives of this talk are indications for anti-sperm antibody testing, review the principle and protocol for the technique by which we do the anti-sperm antibody test, which is the mixed antiglobulin reaction. And in short, it's called the MAR test. Learn scoring, calculation, and reporting of the results by this test, and recognize the importance of quality control, quality assurance, and troubleshooting. Next slide, please. So this slide illustrates the indications for sperm MAR test. So the most important indication is sperm agglutination, which could be mild, moderate, or severe. And on the right side, you can see on the illustration the presence of sperm that are agglutinated to each other. The second is low total motility, low progressive motility, vasectomy reversal. Any disruption in the barrier of the blood-testis barrier can result in formation of sperm antibodies. Scrotal injury, which again leads to disruption of the barrier, previous miscarriages, and poor IVF outcomes. Next slide. So the equipment and supplies that are needed for conducting this test are the bright field microscope using the 20x magnification, phase contrast, and with the green filter. The others are what are listed are the incubator and some other supplies. And finally, what is needed is a fixed cell sperm counting chamber and a humidified chamber in which the samples are incubated. Next slide. So the reagents listed are, and we have provided the illustrations on the right side. The first important reagent is the MAR test Igg kit. And this kit is illustrated as the first illustration, and it is labeled. So this has initially the suspension of the polystyrene latex beads, which are 2 micrometers in diameter, and these are coated with the human Igg. And then there is the MAR anti-serum, which is containing the monoclonal antibodies towards the fragment C of the human Igg. The second important reagent is the controls, which are the positive and the negative controls. And these are run in the assay to make sure that the assay is run correctly. The third illustration on the top is the Igg kit. So this Igg kit has the latex beads and the anti-serum in a single solution. And you can see our technologist at the bottom. She's performing the sperm MAR test. Next slide. What is the principle of the MAR test? Essentially, there are 3 immunoglobulin classes, IgA, IgG, and IgM. But in the semen sample, the main antibodies are IgA and IgG. And what leads to the antibody production is the autoantigenic properties of the spermatozoa. And on the right side, you see the illustration where the beads are attached to the spermatozoa and the beads are coated with the antibody to the IgA. And these will attach to the sperm-bound IgA antibodies. So basically, there is a bridging antibody which is connecting the beads to the spermatozoa. And the motile spermatozoa and the beads will form mixed agglutinates, and that's why it's called a mixed antiglobulin reaction. And it can be tested against both IgA and IgG antibodies. And the binding of the beads indicate the presence of IgA or IgG antibodies. Next slide. So here I'll show you a short video clip of how the test is performed, the IgG test. And you can see there's a humidified chamber. So the slide is labeled, the semen sample, 10 microliters is placed on the slide. Next, we place the beads from the IgG, 10 microliters, and then the semen and this beads are mixed well. Next, we take the anti-serum 10 microliters and add it to the mixture, and then mix the sperm sample, the beads, and the serum. And then a cover slip is placed on the slide. This slide is placed in the humidified chamber. We place a timer for 3 minutes. So once the incubation is done, the reaction has happened, then this slide will be examined on the phase contrast microscope. Next slide, please. So what is the significance of positive IgA and IgG antibodies, which are both tested in the semen sample? The IgA antibodies have greater clinical importance than IgG antibodies. Essentially, the beads will bind to the motile and immotile spermatozoa, which have the surface-bound antibodies. And this is illustrated in a very nice illustration on the right side, the binding of the beads to either the head or multiple sites of the spermatozoa where they are bound, and the percentage of motile spermatozoa with the bound beads is recorded. So the important thing to understand is just count the motile spermatozoa with the bound beads. Next slide. Here, what are the quality reagents? The quality control reagents are basically decomplemented patient serum, which is diluted in cell culture medium, and both positive control and negative control is provided with the kit. These are warm to room temperature before use, and the shelf life is about 18 months. Next slide, please. So here I've provided a step-by-step protocol of how the MAR IgG positive and negative controls are run. So the first step is to vortex both the controls well before use. And the second step is we use a known negative. Known negative means this donor sample. It's a normospermic subject, and his sample was already tested for antibody and was negative. And this sample, to this sample, we add modified human tubular fluid, twice the sample volume. And once we have added the HTF, this is mixed well, the step 4, and then this sample is spun for 10 minutes at 1600 RPM. And once the centrifugation is done, the 5th step is to remove the supernatant and leave some of the supernatant, about 100 microliters, to mix the pellet. And the last step is used, washed donor sample is used to test the negative and the positive controls. And this I will illustrate in a video clip in the next slide. Next slide, please. So here, the semen sample from the donor is there, and then modified HTF is added, twice the volume. This mixture is placed in the centrifuge for 10 minutes. The aspiration of the supernatant is done. The pellet is left undisturbed. And then the pellet is reconstituted with some more fresh modified HTF, and the concentration should not be higher than 50 million. So this is the positive control. 50 microliters of the positive control is added to the amber-colored vial, and then the donor sample is added 50 microliters. Same is done for the negative control. And then once the mixture is in the vial, these vials will be incubated for 1 hour for the reaction to happen. And then the actual testing will be done with the IgG beads as well as the IgG anti-serum to check whether the positive ran positive or the negative ran negative. Next slide. So to examine the sperm MAR test scoring, what are the basic principles? The latex beads bind to the spermatozoa with antibodies on their surface, and there could be few beads or multiple beads that may attach to the motile spermatozoa. So as a result of the binding of the beads, the motility is reduced, and especially when there are large clumps of beads binding to the spermatozoa. The sperm which do not have the antibody coating will swim freely between the beads. So we will count only motile spermatozoa, and a total of 100 is counted, the ones that are attached versus the ones where there is no bead attachment. The ultimate goal of this test is to evaluate the percentage of motile spermatozoa with the beads attached. Next slide, please. So going further, how do we read the results? We use the 20x objective, as you can appreciate in the illustration on the right, and use the phase contrast with the green filter. And then count 100 motile spermatozoa twice to assess the percentage of sperm binding to the beads. What you report is the average of the two values you have obtained, and the test is reported as positive if greater than or equal to 40% of the sperm have beads attached. And this is per the Igg Kit, the manufacturer recommendations, that any test where it is greater than or equal to 40% binding should be reported positive. Now, as per a publication from Human Reproduction from Lotti et al., they have reported a prevalence of MAR test positivity in the 5 to 15% of infertile males. So it is an important cause of infertility. Next slide, please. Here I have an illustration of a negative control sample. So you could play the video. And what I want to show is basically there are beads, clumps of beads, but the spermatozoa are predominantly swimming or moving freely, so they are not bound to the beads. And so we have the negative control run as a negative, and we know the test is performed accurately. Next slide, please. Oh, there was another one. Please go back. The positive control. So here you will appreciate the positive control. And what I want to show you here is the clump of beads and the spermatozoa that are attached in this clump, and they're attached either by the tail or by the head, but they are basically bound to the beads. And so this was a positive control. Reporting of results, negative results are reported if less than 40% of the sperm is bound to the beads. Positive results, how do we report and record? Positive if greater than or equal to 40% of the sperm is bound to the beads. We report the percent binding, and we report the area of bead attachment, whether the head, mid-piece, because if it's bound to the head, then the clinicians have to make a separate judgment call. And on the right, you can see and appreciate the different areas where the sperm is bound. The first one shows the binding to the head. The second shows to the tail. At the bottom is a negative result where there is sperm that is swimming and there's no binding. Next slide, please. So quality control is an important component of the anti-sperm antibody testing. And in our laboratory or other laboratories perform the proficiency testing. So this is an example of two analytes. We are sent two analytes, and the numbers are provided 17 and 18 by the College of American Pathologists. So the first analyte with the red box shows 4% binding. It is negative. The second analyte shows 87% binding, and it is positive. And then the negative and the positive controls results are also shown. The positive control, 97% positive binding. Negative control, there was 13% negative binding. So we know the results are accurate, and this is what we report as part of EQC. Next. So these are the results for both the analytes, and the intended response matched what our results were. So the lab is performing the test accurately and reporting the results well. Next slide. So here is a scenario for a negative ASA IgA test. And this was an IgA ASA, or the anti-sperm antibody test, performed for a sample with sperm concentration of 15 million, motility of 45%. How do we calculate the percent total binding? It's number of sperm with bound beads upon total number of sperms counted and reported as a percent. So when we examine under the microscope, what we saw was free total motile 75, bound total motile 25. So the percent total binding is 25%, and the result is ASA IgA test negative. Remember, the cutoff is greater than equal to 40%. Next slide. This is scenario 2 that is illustrating a semen sample with low motility. So this was a semen sample with 5% motility. And these are the patients where, anti-sperm antibody is ordered by the physicians, and a concentration here was 2 million. When the sample was examined under the microscope, there were less than 100 motile sperm on the wet prep. Percent binding cannot be reported due to very few motile sperm, and hence the result was not reported to the clinician because of very low motility. Next slide. So this is a recent study published in Human Reproduction, which examined the significance of positive MAR tests. So on the first panel, the bar diagram shows percent of men with MAR test positivity. They took a cutoff of greater than 50%, and this was examined in males of fertile couples versus male of infertile couples. And you can appreciate that there was significantly higher number of positivity in males of infertile couples. The second bar diagram shows comparison of positive MAR test and negative MAR test in men with history of epididymitis. Epididymitis is, again, there is a disruption of the blood-testis barrier, and what they have reported is a higher percent of MAR test positivity in patients with epididymitis. So it can be positive in patients with orchitis as well as in patients with epididymitis. Next slide. So I would like to sum up with some take-home key points. MAR test is useful in the diagnosis of immunological infertility. It is important to test for both antibodies, IgA and IgG. And anti-sperm antibodies do play an important role in male infertility, as has been demonstrated by several publications. And proficiency testing, or EQC, is an important tool for reporting accurate anti-sperm antibody results. On the right, you see our andrological evaluation of male infertility. This has the protocol for the MAR test provided in it. Next slide, please. So this is our group in front of the Andrology Center. And I thank you all for your attention.
Let me introduce our next speaker, which is Dr. Sharma. And Dr. Sharma is going to be speaking again. His topic today is on a special test for Isospermia. This is a hidden gem. I am sure that many of you may not be totally familiar with this kind of test. So as as a clinician or a laboratorian or a reproductive professional, you will really enjoy a test which has been used and developed by our program many years ago. So, Rakesh, you can begin the presentation.
Thank you, Dr. Agarwal. Today I will talk on the special test for Isospermia. Next slide, please. The learning objectives are when to perform this test, which is also called the nuclear fast picro indigo carmine test, or in short, Nfp staining test. How to rule out the sperm in the wet prep. Understand the principle of the cytospin involved before the slides are stained. Review the Nfp staining steps. Understand the quality control and the reporting of the results. And in the end, I will leave you with some take-home message. Next slide, please. The incidence of azoospermia is 10% in the infertile men. However, the semen analysis is unable to identify the samples which are azoospermic. Our group developed and published a special test called the Nfp test for azoospermia, and this was validated and published in 1998. The image on the left shows the steps involved, and these are: the sample is examined on a routine semen analysis. If we do not find any sperm on the neat sample, the sample is centrifuged and examined. If no sperm are seen, the subsequent steps will be followed. But if sperm are observed in the spun sample, then this is a case of Cryptozoo Spermia or hidden sperm. When no sperm are seen on the spun sample, then we proceed for the special test, which is the Nfp staining, and this is preceded by the staining and the cytospin step. And when the slides are stained, we will either see the sperm or we will not see sperm, and this is how we will validate the azoospermia test. The image on the right illustrates the protocol for the Isospermic test, and this has been shared with all of you. Next slide, please. So before the smears can be prepared, they have to be processed using the cytospin, and cytospin is a benchtop special centrifuge. It has specially designed rotors and sample chambers. The cells are deposited on a very clear defined area on the glass slide, and the cytofunnel is used, and this cytofunnel has a filter card which actually absorbs the excess of the fluid so that the cells are flattened in a small area on the slide, and the rare spermatozoa can then be identified by this test. On to the right, you see the cytospin and the inside of the cytospin. Next slide, please. The equipment and material required is the cytospin centrifuge, the cytofunnel, and the microscope. Next slide, please. The steps involved in the cytospin are: the sample is mixed and the slide is placed on the slide holder. The cytofunnel is placed on the metal slide holder, and a drop of semen sample is added. And this is followed by a drop of saline sample. Next slide, please. Here I would like to show you a short video clip, and the patient information is provided on the slide, and the slide is placed in the metal holder. You can see the small circle on the metal holder. That is where the slide sample will be followed by the cytofunnel. The cytofunnel is placed in the metal holder and placed in the rotor. The sample is well mixed, and a drop of the sample is added onto the cytofunnel. We also include a blank slide which will not contain the semen sample but will only contain the saline, and this slide is used as a quality control. Once the samples are placed, these are capped. Here you can see we have added a drop of saline and the samples are capped. The rotor lid is placed and the sample is spun to allow the sample to be deposited on the slide. Next slide, please. The Nfp staining allows to identify the rare sperm when no sperm are seen during the initial semen analysis. And these spermatozoa can be identified using a bright field microscope, and the sperm heads are stained red and the tails stained green in this Nfp stain. Next slide, please. The reagents and supplies used for Nfp is shown in this slide. Next slide. The preparation of the stain involves, number one, the nuclear fast stain is a red stain, and this is prepared by dissolving 5 grams of aluminum sulfate in hot water, to which 0.1 gram of nuclear fast red dye is added, and this stain is stable for 6 to 8 months at room temperature. To prepare the picro-indigo-carmine stain, 150 ml of saturated picric acid solution is added in a glass beaker, to which 0.5 grams of indigo carmine dye is added. This dye is light sensitive and the stain should be prepared and stored in a brown or amber colored bottle. And this stain is also stable for 6 to 8 months. Next slide, please. Once the cytospin slides have been prepared, they are fixed, and the second step is the staining using the nuclear fast stain. This is done for 15 minutes, followed by a quick staining with picro indigo carmine. Next slide, please. Here I would like to show you the video clip showing how the staining is done. The smears are fixed and air dried. And the first step is to add the nuclear fast stain. The slides are placed flat on a slide holder, and the slides are flooded with the nuclear fast stain and stained for 15 minutes. At the end of the staining time, we drain the excess of the stain and allow the slides to be placed vertically. And this is followed by the next step, which is the staining with the indigo carmine. The slides are again placed on the slide holder and we flood the slides with indigo carmine. And this staining is done very briefly for about 30 seconds. At the end of the staining, the excess of the stain is drained and the slides are next washed with ethanol to remove the excess of the stain. The slides are allowed to air dry and then they will be cover slipped and ready to be observed under a bright field microscope. Next slide, please. For the microscope setup, we use a bright field, a 100x objective and a 10x eyepiece. We also use the immersion oil for clear visibility of the stain slide. Next slide. The Nfp staining results will include a quality control slide, which is prepared from a normal semen sample and stained along with the Nfp slides. The results are reported either as no sperm seen or the sperm are seen, and the image on the right shows how the Nfp slide is stained. The sperm head stains pink and the tails are stained green. So it is easy to identify the sperm using this Nfp stain. Next slide, please. The quality control for this test is very important, and the quality control is done at two stages. One is during the preparation of the smear, and along with the test sample, we include a blank slide, and the blank slide contains only the 2 drops of saline, and this is used to check for any contamination. The second quality control is used at the time of the Nfp staining, and this includes a smear from a normal semen sample to check the quality of the stain. And we perform a visual monthly check for the contamination of the working components. Next slide, please. This test was developed in our lab and validated. And in this slide, I would like to share how the nuclear fast stain allows to identify the sperm in non-seen samples or samples where no sperm are seen on a routine test. As you can see, the spermatozoa were identified in the samples illustrated on the box to the right, and the positive cells were also identified. And when these were compared with the histological or the clinical diagnosis, there was a good match of finding spermatozoa in 45% of the cells, which did not show any sperm on the initial semen analysis. So this is a test that can be used in an andrology lab to identify and confirm Isospermia in the samples which do not show any sperm on a wet prep. Next slide, please. So the take-home message is that even though the incidence of non-obstructive azoospermia in infertile men is 10%, it cannot be confirmed on a simple routine semen analysis. Our lab has developed a test that confirms the diagnosis of azoospermia in a given sample, and the Nfp stain identifies the presence or absence of sperm in the sample. The identification of the sperm on special staining, it allows the clinicians to select azoospermic patients who have the highest chances of finding sperm during the surgical sperm retrieval techniques. Next slide, please. In the end, I would like to share this group picture, and thank you very much for your attention.
So the next presentation is going to be again by my associate, Dr. Sejal Gupta, and she's going to be speaking about a very common but very important test called post-vasectomy semen analysis. So again, I think this is a very common test because vasectomy is one of the most common method of contraception, at least in the United States and in many parts of the world. So to really to provide the clarity to the patient when the procedure for vasectomy has been performed, when they are cleared of any type of spermatozoa, this test is very critical. But there is a lot more than you may know about this test. This is what we are going to talk about today. So let me stop my video and myself and let Dr. Gupta present her talk now.
Thank you very much, Dr. Agarwal. Previous slide, please. So good morning once again. The topic of my presentation is post-vasectomy semen analysis. Next slide, please. The learning objectives. Firstly, the indications for the test. Then learn how to do post-vasectomy semen analysis. Review the AUA or the American Urological Association guidelines and reporting of results. Also learn the importance of external and internal quality control, as well as quality assurance associated with this test. Next slide. So here, this slide provides the post-vasectomy semen analysis workflow, or the step-by-step procedure of a post-vasectomy semen analysis. The test is performed 3 to 4 months after the vasectomy procedure, and the patient is provided instructions for specimen collection. This could be collection at a lab where they are provided instructions about maintaining the abstinence time of 2 to 7 days. If they're collecting at home, then instructions to collect after the appropriate abstinence time, collect in a sterile container, and transport the sample within an hour and keep the sample at body temperature. And then the performance of the semen analysis. So on the right side, you see the phase contrast microscope, which is used. And at the bottom is provided the semen analysis before vasectomy, after 3 months, where majority of the times sperm will not be seen, but in some samples sperm is seen, and we will describe these scenarios. And after 6 months of the vasectomy, no sperm will be seen. So the reporting of results will also be important. Next. The equipment and supplies, I have illustrated the bright field microscope, phase contrast, green filter that is used, and then the various other supplies for semen analysis, and of course the fixed cell sperm counting chamber. On the right side, you see Dr. Agarwal looking at a semen analysis from a post-vasectomy sample. Next slide. So the stepwise protocol is a post-vasectomy semen analysis includes examining the volume of the sample, preparing a wet prep, and estimation of round cells. The finding of no sperm seen should be reported after complete screening of 2 wet prep slides. And these wet prep slides are prepared on a fixed cell chamber. There are 2 wells, so you could simultaneously aliquot the sample on the 2 chambers and examine them. A definite report of no sperm seen can be made exclusively from wet prep without the need to examine the spun sample. So this is a very key takeaway, that there is no need to examine the spun sample. Next slide. Pvsa Guidelines. So these are the American Urological Association guidelines, and they've provided the definitions for post-vasectomy semen analysis. The first is Isospermia, where there is absence of sperm during microscopic examination of at least 50 high-power fields in a single, well-mixed, uncentrifuged semen sample. The second, rare non-motile sperm, also abbreviated as RNMS, is presence of less than 100,000 non-motile sperm per ml, again based on similar microscopic examination as the first one. And then recanalization is a histologic diagnosis, which they have provided the definition. Next slide, please. The post-vasectomy results categorized based upon the AUA guidelines. So these are 5 scenarios I have provided. And the first and the second, if you examine the first one, the sperm concentration was greater than 100,000 non-motile sperm per ml. And this does not meet the AUA guidelines. Whereas the second one, in contrast, where the sperm concentration was less than 100,000 non-motile sperm ml, meets the AUA guideline, and the test will pass the AUA criteria. Similarly, the third scenario, less than 100,000 per ml sperm, meets the AUA criteria. Whereas scenario 4 and 5, there's presence of a rare motile sperm, which will not meet the criteria. And fifth also has a rare motile sperm, which makes it not meet the AUA guidelines criteria. And these tests have failed the AUA guidelines. Next. So this is a recent study from last year where the study examined the post-vasectomy outcomes and the follow-up of these patients. Are there any patients that are lost to follow-up? So this was a big study where more than 4,000 men were examined, and this was conducted at the Mayo Clinic. And the first bar diagram shows the initial, which is the first post-vasectomy semen analysis results. So majority of the men passed the test per the AUA criteria, either had Isospermia or rare non-motile sperm, the blue and the green figures. But look at the right side. In the red box is the men who failed the AUA criteria. Either they had greater than 100,000 non-motile sperm per ml, or they had presence of motile sperm. So 14% of the men failed the first Pvsa. Now at the bottom, the significant findings are listed. So what is important to appreciate here is that 61% of the men with failed first Pvsa did not get a second post-vasectomy semen analysis. So there is a loss to follow up, and the clinicians need to get to these men and make sure that they do the second Pvsa as well. On the right side is the results of this repeat post-vasectomy semen analysis in 163 men. Majority, again, passed the AUA criteria guidelines, but 32.5% of the men, as seen in the red text box, failed because they had greater than 100,000 non-motile sperm. So these men will require a repeat Pvsa and continued follow-up to make sure that these men are appropriately counseled and followed up. Next slide. So these are some more results. These are actual results, but we have anonymized, and these provide the results per the AUA guidelines. So if you look at the first three results, they have failed the AUA criteria. The first one had a concentration of 340,000 non-motile sperm, plus had rare motile sperm seen. The second one, 2.4 million with motile sperm, fails the criteria. If you examine the last two sample results, you see that they have passed because sample number 4 had Isospermia, sperm concentration of 0.0, and the last sample had non-motile sperm less than 100,000 per ml. So the criteria is to have less than 100,000 non-motile sperm, and this will pass the test per the AUA guidelines. Next slide. Internal and external quality control is important for any test, and this is performed in all andrology laboratories. So the internal quality control is testing the quality of the fixed cell chamber, which can be done by doing the low control and the high control ACU beads, or the latex QC beads are tested each day of testing. And once they are within the reference range and have met the quality control and have been reviewed, then the lab is ready to do the test. The bottom diagram shows the external quality control, and this is results of the post-vasectomy sperm count, 2 analytes, which are sent by an EQC agency. And the first box with the text box with the red outline shows the presence of sperm. And at the bottom is the concentration, which is 2.30 million per ml, and this failed the AUA guidelines. So this is what I wanted to highlight. The second sample, which is PVR04, in the second red text box shows absence of sperm. So no sperm were identified, and this sample had 0.0 sperm concentration or Isospermia. So this sample passed the AUA criteria, and the sample was negative on testing. Next slide, please. The overall take-home message is that post-vasectomy semen analysis helps confirm the success or failure of vasectomy. Pvsa monitoring or repeat vasectomy may be considered by the clinicians if the results show more than 100,000 non-motile sperm per ml or any motile sperm. And so they need to be continually monitored, and the decision needs to be made by the clinician. The last take-home message is that the couple can be advised to stop other methods of contraception if the test shows less than equal to 100,000 non-motile sperm per ml. So thank you very much. And this is my presentation on retrograde sperm count. Next slide, please. The learning objectives of this talk are indications to perform the retrograde sperm count and understanding the special conditions for specimen collection, because 2 specimens will be collected here, learn the procedure and reporting of the results, and reviewing the quality control and quality assurance, as well as troubleshooting associated with this test. Next slide. Indications for the test are, firstly, Isospermia. The lack of presence of sperm is an important indication for performing this test. Second is low semen volume. Thirdly, Aspermia. So lack of antegrade ejaculation or lack of semen expulsion from the urethra or a dry ejaculate is an important indication. Low sperm concentration. And the last is urological surgery for benign prostatic hyperplasia is known to lead to retrograde ejaculation. On the right side is the phase contrast microscope. The equipments and reagents. Next, please. The most important equipment is the centrifuge, as shown and on the right side, and then the 2 specimen containers that are labeled with semen and urine. So this makes it clear to the patient that, how he has to collect the semen in the semen container and the urine in the urine container. And the third important component is sperm wash media, which is human tubular fluid. And this is what the urine sample has to be collected in, and the other supplies are also listed. Next. So retrograde sperm, the specimen collection. So the first and the most important instruction to be given to the patient is that they will need to collect at the lab. And when the patient is in the lab, the important step is to instruct the patient to urinate without completely emptying the bladder. So partial urination followed by collection of the ejaculate, which is collected in the container marked semen, and then he will collect a semen sample post the orgasm and the ejaculation in this container, which is marked urine and has the pink HTF, which is 9 ml of warmed up HTF, which was kept in the incubator for 20 minutes. Next, please. So here is the retrograde sperm count protocol, the step-by-step procedure. After the specimen collection, the semen sample is placed in the incubator for liquefaction, and at the same time, start the analysis of the urine sample. Before analysis, measure the volume of the urine plus the HTF mixture. This needs to be documented. And on the right side are the 15 ml centrifuge tubes with the urine and HTF mixture. Next slide. So post the transfer of the urine sample into the conical tubes, these tubes are placed in a centrifuge, and then they are centrifuged at 1600 RPM for 10 minutes. And after the centrifugation, next step, next slide, please. The centrifugation has been done, and on the right side is the illustration with the sperm pellet at the bottom. So the supernatant is going to be aspirated, and make sure not to disturb the pellet or the cloudy portion. This supernatant, once it's aspirated, the pellet is reconstituted in 1 to 2 ml of HTF. And finally, note the total volume of the reconstituted sample. So each of the pellet from each tube is reconstituted in fresh HTF and finally pooled together, and the total volume of the pooled sample is recorded. Next slide. And the final total pool volume of the well-mixed urine sample, one would take an aliquot of 6 microliters and load this on a fixed cell counting chamber. So this is examined under the phase contrast microscope. The first step is to look for the presence or absence of sperm, and then if sperm is present, estimate sperm count and motility. The important point to understand here is the final reconstituted urine volume is used for calculation of sperm concentration and total sperm count, which are both reported. And on the right side is an illustration with a centrifugation and aspiration of the sperm pellet. Next. So here I wanted to highlight a study by a group, Mehta et al., and they have examined the significance of post-ejaculatory urine analysis in infertile versus normal men. So the salient findings of the study were, and they had equal numbers of like 70 infertile men versus 70 normal men with a normal semen sample. And the first important finding of the study was that the sperm were found in post-ejaculatory urine in higher numbers in infertile men compared to the controls, and this was significantly higher. And this is illustrated in figure 1 on the right side. And it is highlighted with the red text box. The significant difference, 24% sperm in controls versus 46% in infertile men. The second finding was that low semen volume is associated with higher percentage of sperm in urine. So this is illustrated in figure 2, the relationship between semen volume and percent total sperm in the urine sample, and infertile men are the black boxes. So this is highlighted with the red circle. With the low volumes or even like no sperm sample being produced, there are a higher percentage of sperm that are found in infertile men versus the controls. Overall, the study reports, which is the third significant finding, that severe oligospermia with low semen volume is directly related with high sperm counts in post-ejaculatory urine samples. Next slide, please. The internal and external quality control associated with this test is mainly to be reporting accurate sperm count and motility. And the red text box in the first illustration shows a normal sample which is examined, and manual count motility is performed, as well as the CASA count motility is performed on the same sample, and there is good correlation between the manual values and the CASA values, establishing internal quality control. The second illustration at the bottom is EQC for the sperm count, and this is a sperm motility video captured on a fixed cell chamber, which is provided to us by the EQC agency. And the red circles highlight the first analyte concentration of 19.2 and the second analyte concentration of 22.2. And then these values are compared with other labs, and our values reported in the lab have to fall within mean plus minus 2 standard deviations of the overall results. And then we meet the EQC criteria and establish that the results from the lab are accurate as well as precise. Next slide. Retrograde ejaculation is associated with infertility, and in these infertile couples, sperm recovery can be done from the post-ejaculatory urine, as shown on the first box, and then regulation of the pH and osmolarity of the urine sample will help improve recovery of the viable sperm. And the recovered sperm can then be used for ART, either IUI or IVF. Next slide. So what is the step-by-step protocol of sperm recovery from retrograde ejaculate for ART use? The retrograde urine sample is centrifuged, the pellet is reconstituted following centrifugation, and this pellet is then layered on the gradient media. A gradient wash is performed, and then the prepared sperm can be used in ART. Next slide. As I alluded to earlier, the regulation of urine pH and osmolarity is important of sperm survival. And this study reported in fertility and sterility looked at normal subjects, 6 in number, and their semen sample was exposed to different types. The first image shows sperm exposed to unprepared urine, which is the first urine sample in the morning with a very low pH and a high osmolarity. And this study examined the change in progressive motility grade A and B once the semen sample was exposed to the urine sample. And as the first figure illustrates, there was a marked reduction, immediate reduction in motility of 42%, ranging up to 80% in the sample exposed to unprepared urine. The second image illustration reports sperm exposed to prepared urine, and this exposure resulted in retention of motility. So these were men who were given orally soda bicarb in the morning, and then the urine sample was having a good pH of 7.5 and a normal osmolarity of 300, as illustrated here, and will result in retention of motility. So optimization of sperm can result from regulating the urinary pH and osmolarity, as is done for sperm recovery from retrograde ejaculates. Next slide. So my take-home key points are that screening of post-ejaculatory urine is necessary in isospermic patients with low semen volume. It's important to observe special collection conditions, including intake of soda bicarb to regulate pH and osmolarity. The collection has to happen in the lab. It has to be, the urine sample has to be collected in sperm wash media. And finally, the retrograde urine sample can be processed for potential use in IUI and IVF. And on the right side, you see the technologist preparing a retrograde urine sample. Next slide. So this is our group in front of the Andrology Center at the Cleveland Clinic, and I thank you all for your attention.
Thank you very much, Sejal, for an excellent presentation. Now we move very quickly to our last presentation, which is the clinical presentation. The speaker here is Dr. Neil Parekh. Dr. Parekh is a clinical assistant professor, a young man in the Department of Urology. He did his fellowship at Cleveland Clinic in Andrology and Male Infertility 2 years ago, and went on to join the staff after he completed it in the summer of 2019. He has a lot of surgical experience in a variety of urological procedures related to microsurgery and male infertility, as well as erectile dysfunction. So Neil has been a very important part of this lecture series that we are doing, as he's going to stitch together all the tests into providing a clinical interpretation of a variety of tests that we have discussed today. We discussed 6 different laboratory tests which are part of the semen analysis: the Leukocytospermia, the Vitality test, the Anti-Sperm Antibody, the special test for Isospermia. We talked about the post-vasectomy semen analysis, and lastly, Dr. Gupta spoke about the retrograde sperm count. So without any further ado, let me mute myself first. Okay, for this. Okay.
Hi everyone, my name is Dr. Neil Parekh. I am a clinical assistant professor in the Department of Urology at the Cleveland Clinic. Today I will be sharing with you the clinical interpretation of laboratory results. By now you should have heard lectures from Dr. Henkel, Dr. Gupta, and Dr. Sharma, and have a better understanding of some of these specialized tests, and we'll discuss how we use them in the clinical situation. Let's start with the first clinical question. We have a patient who is being evaluated for infertility. He has a semen culture and semen analysis performed. The report shows a positive semen culture. However, it is negative for Leukocytospermia. What is the management strategy? Now, this is something that we try to avoid on a routine basis, but does commonly occur. We try to avoid a positive semen culture in the setting of negative Leukocytospermia by adhering to the strict hygiene protocol that the WHO recommends. Furthermore, we recommend that patients urinate before they provide the semen analysis. Unfortunately, skin flora contamination may be unavoidable, as we see in this situation. Contamination can be seen because as much as 71% of the bacterial strains colonizing the coronal sulcus of the penis are also present in the distal urethra. There is also significant controversy regarding the role and utility of semen culture. It has been shown that no significant difference exists within the presence or absence of bacteriospermia in achieving pregnancy. And there are no strict guidelines that revolve around the management of semen cultures. However, in my clinical practice, I do believe a positive semen culture with Leukocytospermia should be treated. Question number 2. We have a patient who has a semen analysis that demonstrates a concentration of 12 million, a motility of 30%, an elevated nth value of 4.5 million WBCs per milliliter, and a negative semen culture. Will this patient benefit from a course of antibiotics? This is a very common clinical scenario that I encounter. I just want to break it down a little further. So this male with Oligostenozoospermia presents with Leukocytospermia. The terminology is also sometimes seen to be pyospermia or Leukospermia. Just to avoid any confusion, you can see it written in any of these ways. Just like any patient who presents to my office, we'll begin with obtaining a thorough history and physical examination. It is also important to obtain social history. This includes smoking, alcohol intake, drug use, etc. The reason this is important is because Leukocytospermia has also been found to be caused due to cigarette smoking, heavy alcohol use, advanced age, and not just infection. So here we have an algorithm where we break down the testing required in this patient. So in this patient with an elevated nth test, and we determine that there are leukocytes present, a semen culture and PCR should be performed. The PCR will allow us to check for gonorrhea, chlamydia, atypical bacteria such as ureaplasma and mycoplasma, and depending on the results of the culture and the PCR, will determine the treatment regimen and whether antibiotics will be of benefit. Now, there's a few different situations that can occur. Of course, in a patient that has been treated with antibiotics but continues to have an elevated nth level after treatment, it is possible that their social history could indicate why they have a persistently elevated nth. So if they're a heavy smoker, we may not see their nths completely normalize. Another important clinical question in this case is, was the partner treated? So an infection such as gonorrhea, chlamydia, ureaplasma, mycoplasma can be transmitted from the male to the female or vice versa. So if you're only treating the male, it is possible for the male to continue to get reinfected if they're still sexually active with their partner. So it is important to document or note whether the female partner has been treated as well. Let's move to clinical question number 3. We have a patient who has a semen analysis that shows normal volume at 2.5 milliliters, normal sperm concentration at 50 million, 0% sperm motility, and a vitality of 40%. So as Dr. Sharma discussed in his lecture on sperm vitality testing, it is indicated in patients with low motility, less than 25%, in either ejaculated or surgically retrieved specimens. From a clinical standpoint, it is important to differentiate between necrozoospermia, which is dead sperm, and astenozoospermia, which is viable immotile sperm. Normal vitality is greater than or equal to 58%. So again, like in these patients, an important history and physical exam is important. So in this particular situation, a history elicits a history of recurrent respiratory infections. So in a male with recurrent respiratory infections and immotile sperm, the likely diagnosis is primary ciliary dyskinesia. This is a rare genetic disease with symptoms caused by malfunction of motile cilia. However, even with completely immotile spermatozoa, the use of the hypoosmotic swelling test is a good alternative to identify which spermatozoa may be viable for IVF ICSI. Question number 4. A patient presents for semen analysis and has the following results: a semen volume of 3.5 mLs, a sperm concentration of 35 million, sperm motility of 0%, and a vitality of 0%. How will you manage this patient? So in this situation, unlike the previous patient, this is complete necrozoospermia. So when a patient presents with this diagnosis, it's important to elucidate whether any spermicidal lubricant was used during the semen analysis collection. You want to make sure there was no issue with the collection process itself or any other issue that could result in the necrozoospermia. It is important also to repeat the semen analysis, just as the AUA guidelines and EUA guidelines recommend one semen analysis spaced out by approximately 1 month. If there is no pathological cause, IVF ICSI has been shown to be successful when using testicular-derived sperm in men with necrozoospermia. Clinical question number 5. We have a patient who presents with primary infertility and with the following semen analysis results: a sperm concentration of 10 million, a motility of 25%, but also moderate to severe sperm agglutination. How will you manage this patient? So another common clinical scenario that I encounter is a patient with oligoastenozoospermia with moderate to severe agglutination. As was discussed in the previous lecture, sperm agglutination refers to sperm being clumped together. This process may impair sperm motility and prevent sperm penetration of the cervical mucus and inhibit sperm oocyte fusion. Now, what can cause moderate to severe agglutination? One common cause is violation of the blood-testis barrier. Any violation of this barrier can predispose men to an immune response to sperm in the form of anti-sperm antibodies. So in this patient scenario, what could cause the severe agglutination and the reduction in motility and concentration? So as we mentioned, anti-sperm antibodies, a history of genital trauma, as well as testicular or scrotal surgery, for example, a history of testicular torsion or orchidopexy for cryptorchidism, history of genital infections, or vitamin C deficiency. Clinical question number 6. A patient has a history of a vasectomy reversal 1 year ago and wants to conceive with a new partner. His semen analysis results are as follows. He has a sperm concentration of 4 million, motility of 30%, and moderate sperm agglutination. How will you manage this patient, and when do you order the anti-sperm antibody test? So it is important to know that the presence and levels of anti-sperm antibodies following vasectomy reversal are inconsistently predictors of whether a couple will be successful in achieving pregnancy. The reason being is that ASAs, or anti-sperm antibodies, are found in between 8 and 21% of men in the general population, and 9 to 36% of infertile patients. However, up to 100% of men after a vasectomy will have anti-sperm antibodies. So this is a male that has a history of a vasectomy followed by a vasectomy reversal. So there's a strong likelihood that he will test positive for anti-sperm antibodies. So really there is no role for testing in this clinical situation. Now, regardless of the cause of anti-sperm antibodies, the treatment is pretty consistent for men who test positive, or in this situation. I commonly will treat them with a low-dose steroid, typically 1 month of a tapering dose, and there are multiple different combinations of steroids and different steroids that can be used. I would then recommend repeating a semen analysis at 6 weeks after treatment with the low-dose steroids to reassess the patient's sperm quality. Question number 7. We have a couple who presents with primary infertility. The male partner's semen analysis is as follows: sperm concentration of 54.8 million, a motility of 31%, moderate to severe sperm agglutination, and no female factor. If the couple choose to undergo intrauterine insemination, what special advice would you give them? This is a patient with isolated Pvsa Spermia. Once again, want to obtain a complete history and physical examination, assess for varicocele or any other pathology. You want to assess for risk factors for anti-sperm antibodies, so any previous surgical history or trauma to the genitalia. Now, the AUA guidelines do recommend testing anti-sperm antibodies when there's isolated Pvsa with normal sperm concentration and agglutination, as seen in this patient. However, ASA is not needed to be tested if sperm are to be used for ICSI, and that is also an AUA guideline. Now, in this situation, the couple may be interested in pursuing intrauterine insemination. The evidence demonstrates that really no special treatment should be taken prior to IUI. The most important thing is properly timing the IUI with the female's LH surge, and this is the first-line treatment. Now, if they have failure of IUI, then there may be a role for pre-treatment of the male with low-dose steroids, as I discussed on the previous slide. Clinical question 8. So we have a patient who presents with a semen volume of 3 mLs. He has no sperm on the wet prep. Upon centrifugation, 5 sperm are seen on wet prep. How should this patient be managed? So this is what we consider Cryptozoo Spermia, also referred to as virtual Isospermia. This is a patient with a very low spermatozoa concentration, less than 1 million in the ejaculate. So here we have an algorithm for this patient. So on the wet prep, there are sperm seen. So this is Cryptozoo Spermia. If no sperm were seen, then you would proceed to more specialized staining, which we'll discuss in the next few slides. So what are causes of Cryptozoo Spermia? Varicoceles, infections, environmental factors, history of malignancy, undescended testicle, endocrine disorders, genetic mutations, previous pelvic surgeries, and certain medications, chemotherapy or radiation therapy can result in this diagnosis. Further workup includes history, physical exam, hormonal elevation, genetic evaluation, checking for karyotype and Y chromosome microdeletion. And in this situation, where 5 sperm are seen, it is also reasonable on a repeat semen analysis to offer sperm cryopreservation. Now, we can't predict whether or not more or less sperm or a similar number of sperm will be seen on subsequent semen analysis, but I do feel in this situation we do not want to waste an opportunity to freeze sperm that could potentially be used for ICSI and avoid a surgical sperm retrieval procedure if sperm are frozen. Question number 9. So a patient presents with a semen volume of 0.5. No sperm are seen on wet, unspun, and spun preparation. Upon special nuclear fast staining, a few spermatozoa are reported. How will you manage this patient? So in a patient with Isospermia, and after the history and physical exam which determines the presence or absence of a vas deferens. In this case, this patient did have palpable vas deferens. The next step is to look at the semen volume. So in this scenario, the volume is 0.5, which is considered to be low. So the next step would be a post-ejaculatory UA to further assess this patient. So in this situation, with the positive staining, this increases the likelihood of there being sperm, whether it is retrograde ejaculation or an obstruction, or in the situation where this was normal volume Isospermia, there is a higher likelihood of finding sperm with a surgical sperm retrieval like microsurgical testicular sperm extraction. So we do use this in clinical practice as far as predicting and being able to counsel the patient on the likelihood of finding viable sperm. Question 10. A patient's semen sample is examined for routine semen analysis. The sample shows normal volume and complete absence of sperm on wet prep. Upon special staining by nuclear fast stain, only 2 sperm heads without tails are reported. Will this patient benefit from sperm retrieval by epididymal aspiration? So in this scenario, 2 sperm heads without tails are reported. This is what we call spermatids with no tails, or acaudate sperm. These sperm are often seen in the setting of necrosis. Now, will this patient benefit from sperm retrieval by epididymal aspiration? While isolated sperm heads can be used for ICSI, whole sperm produce a higher fertilization rate. Therefore, in this particular patient, I would recommend the gold standard procedure, which is microsurgical testicular sperm extraction, in an effort to find whole sperm within the testicle. Epididymal aspiration is more useful in the setting of obstruction. So in a patient with a history of vasectomy or some other sort of urologic surgery, hernia surgery, where there's concern for obstruction of the vas deferens, and by aspirating from the epididymos, more likely you would find healthy, viable sperm that could be used for IVF ICSI. However, in this situation, I would recommend microtesting in an effort to find whole sperm. Question 11. We have a 36-year-old male with a history of type 1 diabetes who presents with primary infertility. There is no female factor. He reports a lack of fluid being expelled with ejaculation. He is unable to produce a semen sample for semen analysis. How will you manage this patient? So this can at times be a very complicated scenario to sort out. But what I first recommend assessing is whether or not this patient has an orgasm. For many men, they aren't sure what the difference is between orgasm and ejaculation. So in this particular patient, he does have pleasure and is able to orgasm. However, there is no fluid being expelled with ejaculation, so he has no ejaculation. So normal orgasm and no or low volume ejaculate, the next step is to assess for post-ejaculatory urinalysis. So the EAU guidelines for retrograde ejaculation are as follows. Total or sometimes partial absence of an antegrade ejaculation is the definition of this. Patients experience a normal or decreased orgasmic sensation except in paraplegics. A post-ejaculatory UA is positive if there's presence of any sperm in patients with Isospermia or Aspernia. And typically, this is defined as a similar or more number of sperm present in patients with low volume ejaculate and Oligospermia. So the patient should have more sperm in the urine than in the ejaculate. So what are some causes of retrograde ejaculation? So some background, anatomically, the bladder neck and the internal sphincter fail to close during ejaculation, and this allows semen to flow along the path of least resistance back into the bladder. So what are some conditions that can cause bladder neck and internal sphincter dysfunction. They can be neurogenic causes. So in this particular patient, a history of type 1 diabetes that may not have been appropriately controlled over the years can lead to diabetic neuropathy, which prevents the bladder neck from closing during ejaculation and prevents the antegrade flow of semen. Spinal cord injury, multiple sclerosis, a history of retroperitoneal lymph node dissection, history of colorectal or anal surgery can all affect the nerves to the bladder. Now, medication is probably one of the most common culprits for retrograde ejaculation that I see in my practice, particularly in the form of alpha blockers. Many men are put on alpha blockers for BPH, such as tamsulosin. Tamsulosin can result in retrograde ejaculation, and all men, especially men in their reproductive years, should be counseled on that risk. According to the FDA, the risk of that side effect of tamsulosin is about 7%. Certain other alpha blockers in a similar class may have more or less risk of that side effect. Antidepressants and antipsychotics can also be culprits. And lastly, issues with the bladder neck itself, and this could be a congenital defect or a history of surgery, such as transurethral resection of the prostate or radical prostatectomy. Moving on to question 12. A couple presents with primary infertility for 2 years, and they are discussing going through IUI cycles. The male partner presents with the following semen analysis and post-ejaculatory UA. He has low volume, 0.3 mLs, with sperm concentration of 2 million and motility of 25%, with normal morphology on his semen analysis. His post-ejaculatory UA is consistent with retrograde ejaculation, per the definition, and has a concentration of 24 million and a motility of 20%. What will be your advice to this couple? So again, the EAU has guidelines in this particular situation. So post-orgasmic urine sperm collection is indicated to be used for assisted reproductive techniques if the following occurs. Any sort of drug treatment is ineffective or not tolerable due to side effects. So there are a variety of medications that we commonly use to attempt to treat a poor closure of the bladder neck. In the United States, we commonly use over-the-counter Sudafed or pseudoephedrine, and this alpha agonist can be used to potentially close the bladder neck during ejaculation to promote antegrade semen flow. However, if this fails, then sperm collection from the post-orgasmic urine can be suggested. Also, if the patient has a history of spinal cord injury. And lastly, if the patient is on certain medications, we discussed tamsulosin as one of them, or other form of drug therapy that is a cause of the retrograde ejaculation and cannot be stopped for any particular reason. So in this clinical scenario, we want to attempt medical therapy first. We want to make sure the patient is not undertaking any medications that could be inducing the retrograde ejaculation. And if all else fails, the next step would be able to use it for intrauterine insemination. Question 13. We have a patient that had a vasectomy 3 months ago. His post-vasectomy semen analysis demonstrates the presence of rare motile sperm. How will you manage this patient? So this is another common clinical scenario. Unfortunately, there are guidelines that direct our treatment. So according to the AUA, we should counsel all patients that a vasectomy should not produce immediate sterility. I counsel all patients after a vasectomy in the office that they should continue to use some form of contraception until we obtain that negative post-vasectomy semen analysis. All patients are counseled that vasectomy does not guarantee or is not 100% effective, and the risk of pregnancy after a vasectomy is roughly 1 in 2,000. I commonly check the the first post-vasectomy semen analysis at 3 months, but anywhere between 8 and 16 weeks after vasectomy is the time range for the first semen analysis. I also commonly recommend patients to at least ejaculate 20 times prior to providing that first post-vasectomy semen analysis. It's not a strict guideline, but is something that I commonly recommend. And finally, a vasectomy should be considered a failure if any motile sperm are seen on post-vasectomy semen analysis at 6 months after vasectomy. So in this clinical scenario, it has only been 3 months. There are rare motile sperm. I would recommend this patient ejaculate more frequently and have him repeat a post-vasectomy semen analysis in 1 month and reassess the concentration. A key thing when dealing with post-vasectomy semen analysis is to look at the trend. So I typically look at more than one if the first one is positive. Fortunately, that is not a common scenario. Question 14. A patient had a vasectomy 6 months ago. Post-vasectomy semen analysis reports presence of over 100,000 non-motile sperm per milliliter at 6 months. How will you manage this patient? So, per the guidelines, patients may stop using other forms of contraception when examination of one well-mixed, uncentrifuged, fresh post-vasectomy semen analysis shows the following: Isospermia, only rare non-motile sperm, or less than 100,000 non-motile sperm per milliliter. So in this situation, the patient has over 100,000 non-motile sperm. So the next step would be to repeat the post-vasectomy semen analysis in 1 month. And once again, looking at trends of serial post-vasectomy semen analysis and clinical judgment should be used to decide whether the vasectomy is a failure and whether or not repeat vasectomy should be considered. Clinical question 15. A patient had a vasectomy roughly 1 year ago. Post-vasectomy semen analysis at 6 months showed less than 100,000 non-motile sperm. However, post-vasectomy semen analysis at 1 year shows 100,000 sperm per milliliter with 5% motility. What will be your advice to the patient? Now, this unfortunately is rare, but is seen. I see this scenario roughly once a year. Commonly, the patient had a vasectomy performed elsewhere and either never had a post-vasectomy semen analysis and failed to follow up, or did, in this situation, have a negative post-vasectomy semen analysis, however, are now presenting with a positive test or causing a pregnancy. So vasectomy failure. So technical failure is characterized by persistently normal or nearly normal motile sperm counts and sperm motility after vasectomy. So this patient who had passed his post-vasectomy semen analysis 6 months would not be considered to have a technical failure because it was negative at that time. However, recanalization should be suspected if motile sperm or rising sperm concentrations are seen after an initial routine post-vasectomy semen analysis has shown non-motile sperm or Isospermia. So that is what we have in this clinical situation, which is recanalization. Recanalization can either be transient or persistent based on the results of serial post-vasectomy semen analysis. It is impossible to know the true incidence of late recanalization because post-vasectomy semen analysis is rarely repeated after a post-vasectomy semen analysis shows Isospermia or rare non-motile sperm. But recanalization is also something that I counsel patients on, the risk of the vas reconnecting basically. While it is very rare, it is something the patient should know of. Now our final question, a patient underwent a vasectomy at an outside clinic. He now presents at 2 years post-vasectomy to the Cleveland Clinic. He states his wife is 8 weeks pregnant. What is your next step in the management of this patient? Again, unfortunate scenario. Hate to receive this phone call, but again, has happened. But it is important to remain calm and make sure you obtain all the information before telling the patient what is the likely cause. So one of the most common causes of this is the patient never obtained a post-vasectomy semen analysis as recommended after the vasectomy. According to the AUA, the compliance rate is roughly only 78%. So a quarter of patients do not get the appropriate semen analysis after the vasectomy to ensure a negative test. Now in this patient, we don't know if he had a negative semen analysis post-vasectomy, but let's say he did. If the semen analysis reveals Isospermia, then the physician should inform him that the pregnancy could be due to transient recanalization. So if we repeat the semen analysis and once again shows Isospermia, however, they are certain his wife is pregnant, then there is a possibility of transient recanalization, which I discussed in the previous clinical question. In fact, a number of case reports have confirmed paternity based on genetic testing, even though the man had had multiple previous semen analysis showing Isospermia. Now, one thing you should also let patients know that genetic analysis to confirm paternity is also an option in this situation, but you should not jump to any conclusions when counseling these patients, and repeat testing and giving them an informed management option is the next appropriate step. So several take-home messages from the lecture today. Let's start with Leukocytospermia. So patients with Leukocytospermia should be assessed for bacterial infection by PCR. We are checking for infections such as gonorrhea, chlamydia, ureaplasma, and mycoplasma. And if positive, the patient and their partner should be appropriately treated with subsequent repeat testing afterwards. The utility of semen culture is controversial, but a positive semen culture and leukocytospermia should be treated. So as we discussed, this is a situation where I do recommend antibiotic treatment. However, in the scenario where you have a positive semen culture but negative leukocytospermia, that is more likely contamination and does not need to be treated. It is important to test for sperm vitality, so you can differentiate between Necrozoo and Astenozoo, as the management of both those conditions is different. With Necrozoo, you're looking towards a surgical sperm retrieval from the testicle to find viable sperm, and with Astenozoo, you have the opportunity to use HOS hypoosmotic swelling testing in order to identify viable sperm that could be used for ICSI. A history of blood testis barrier evaluation may predispose a patient to anti-sperm antibody formation. This again is a very important past medical history that you should obtain from the patient. Any sort of surgery or trauma to the testicle can result in severe sperm agglutination due to anti-sperm antibody and should be assessed for. However, in the situation of vasectomy or a man with vasectomy reversal, ASA testing is not required, as there likely are anti-sperm antibodies due to the violation of the vas deferens and testicle during the surgery, and the treatment is a 1-month course of steroids. Now, in a patient, Isospermic male who is positive for the nuclear fast stain, this can help predict the success of surgical sperm extraction, and is something I use on counseling patients prior to surgery. As the surgery is a significant undertaking, there is significant financial burden on the patient. There is also an emotional strain for the patient and the couple as far as whether or not sperm is found. But having this stain be positive can at least help guide the patient in making a educated decision. Post-ejaculatory urinalysis is indicated in patients with normal orgasm and low or no ejaculate volume. It is also important to obtain a good history, whether they're diabetic, have a neurologic condition, or previous surgery to the bladder, neck, prostate, etc. And lastly, when it comes to vasectomy, compliance is low as far as obtaining a post-vasectomy semen analysis after vasectomy. However, only one post-vasectomy semen analysis is needed. A negative test can show either Isospermia, only rare non-motile sperm, or less than 100,000 non-motile sperm per milliliter. That concludes the clinical lecture for today. I hope you found today's lecture valuable. Please don't hesitate to reach out. My email address is parekh, P-A-R-E-K-H, n3@ccf.org. I'm happy to address any questions you may have. Thank you.
This is my concluding remarks. To summarize everything that we heard today in this module, which was focused on ancillary semen test, the peroxidase staining by ENS test, which was covered by Professor Ralph Henkel in his lecture on Leukocytospermia. Even though it's not a gold standard, it is simple, easy, and provides most reliable results. So we definitely think this is something that can be done by most laboratories. It is certainly very inexpensive test. Leukocytospermia may indicate an underlying male accessory gland infection or MAGI, but it is not a good predictor of positive semen culture, as indicated also by Neil. But if it is both of them are positive, then they should be treated. Vitality, which was covered by Dr. Sharma, can be examined by eosin nigrosin staining in specimens with poor motility. It is an important measure of sperm fertility, and it can serve as a surrogate marker of sperm DNA integrity. So certainly there is some evidence that this is something that can be used in laboratories which don't have access to sperm DNA testing. Viability, on the other hand, can be examined by the hypoosmotic spelling test, and viable sperm which are identified by this test can be used in ICSI, because we are not doing any kind of staining. So you can simply select that sperm under the microscope and use it for injecting into the oocyte. The mixed antiglobulin reaction test, or a MAR test, is useful, as explained by Dr. Gupta, in the diagnosis of immunological infertility, which can range anywhere between 8 to 30%, and it is important to test for both IgA and IgG antibodies. The history of blood testis barrier violation may predispose a patient to the formation of anti-sperm antibodies. Next, the Nfp special staining, as discussed by Dr. Sharma, can confirm the diagnosis of Isospermia in a semen sample, as explained by Dr. Neil, and the identification of a sperm on a special staining helps him select Isospermic patient with the highest chances of finding a sperm during surgical sperm retrieval. So this is an important test, and all of you have been provided every single protocol beforehand, which are available on Dropbox for you to read. Next is the test on post-vasectomy screen. This was covered by Dr. Gupta. It can help confirm the success or failure of vasectomy, and something this is very important, where you heard various scenarios from Dr. Neil. Screening of post-ejaculatory urine is necessary in Isospermic patient with low semen volume, and retrograde urine sample can be prepared for sperm retrieval and for potential use in intrauterine insemination and conventional in vitro fertilization. So we heard these various things. The next, excited to discuss the next program which is going to be in the beginning of March rather than February. We are going to space the subsequent presentation by 2 months, just because we found that the preparation of these lectures take enormous amount of time. We cataloged almost 200 hours in preparing our team for today's presentation. It is in addition to our very hectic schedule. It is not sustainable, so we decided we are going to space it, and I believe that should still be extremely well planned and very well prepared. So the next presentation will be on advanced andrology techniques, and we are going to give this on March 9. And the learning objectives will be advanced sperm function test, sperm DNA fragmentation, measurement of oxidation reduction potential, lab developed test for sperm function, and clinical interpretation. And this is the program which is going to essentially cover many of these topics in the most thorough manner and in a very concise platform that we have developed. And the subsequent slate of program has been already planned now. After this presentation in March, we are going to go into the embryology related topics starting in the beginning of May on IVF, ICSI, and micromanipulation. Then it will be on a monthly frequency. Actually, bi-monthly frequency, it will be July that we will have oocyte and embryo handling. In September, embryo biopsy. November, pre-implantation genetic testing. In January of 2022. It seems like a long time, but time will fly. We are going to cover the embryo cryopreservation. In March, vitrification of oocyte, blastocyst, embryo, and sperm. In May, we have ART sperm preparation. In July, cryopreservation. In September, we are going to talk about management of IVF and andrology lab. And in November of next year, we are going to talk about preparing the CAP laboratory for College of American accreditation and FDA inspection. The embryology related topics starting from module 5A all the way until actually module 6 until March of 2022 are going to be covered by our team of clinical embryologists who are really extremely well proficient and are extremely smart and clever. They are going to be covering these topics in coordination with our team. So I want to thank our speakers, Professor Henkel, Dr. Sejal Gupta, Dr. Rakesh Sharma, Dr. Neil Parekh for extremely well-planned and wonderful presentation, which covered all these 6 topics in wonderful detail. Our program coordinators who really managed this entire experience for you right from the day 1 all the way to today. They spent numerous amount of time planning, organizing, and managing everything. It's a lot of work and a lot of thanks to each and every one for their commitment and their help.